Okay, so now moving on to the second line of defense of our innate or internal defense system. When we're talking about the second line of defense, it's predominantly just going to be cells and chemicals. So things like phagocytic cells, natural killer cells, antimicrobial proteins, fever, and even inflammation all contribute to the second line of defense. We're going to talk about these individually here shortly. Many second line of defense cells, they have what we call pattern recognition receptors, and these are going to help recognize and bind tightly to structures on microbes, and they're actually able to disarm them before they can cause any harm. So one type of pattern recognition receptor are what we call toll-like receptors, and these play a huge role in triggering our immune response. We have about 11 different types of TLRs, and each of these can recognize a particular class of attaching microbe. So not just macrophages have toll-like receptors, actually a variety of cells, including epithelial cells lining our GI tract and our respiratory tract, also have TLRs. And TLRs really just allow us to help recognize invaders and then sound an alarm that is eventually going to initiate inflammation. So let's move on to our first cell, and those are phagocytic cells. And if you remember from our blood chapter 17, we mentioned neutrophils. So neutrophils are one of the phagocytic cells that play a role in the second line of defense. They were our most abundant phagocyte, as well as our most abundant leukocyte. The downside to these neutrophils is that they pretty much die fighting, and they become phagocytic on exposure to any type of infectious material. And again, they're going to destroy themselves in the process. When we're talking about phagocytes, because there's another type of phagocytic cell, are basically just our two white blood cells that are capable of ingesting and digesting any type of foreign microbe that we have. So neutrophils was one. And our second one would be macrophages. And if you remember from our blood chapter, our macrophages actually develop from our monocytes. In our monocytes, they were able to leave the bloodstream. And when they leave the bloodstream and enter our tissues, that's where they're going to actually develop into much larger cells that we call macrophages. We typically classify our macrophages into two different categories. One would be free macrophages and the other one's fixed macrophages. Our free macrophages are free to wander throughout all the tissue spaces, and they keep wandering until they come or encounter some sort of debris or foreign invader. Our fixed macrophages are just that. They're fixed. They're permanent residents of a specific organ. So for example, our stellate macrophages are always going to reside in our liver. So when we're talking about our two phagocytic cells, again, our macrophages and our neutrophils, the way they kill these microbes is by a process called phagocytosis. And it has a couple different steps, but the overall goal is to basically bring in the microbe into the macrophage itself. The phagocyte itself, and basically digest it and break it apart and kill it. So phagocytosis is pretty much the process is going to start when our phagocyte is going to recognize or adhere to some sort of pathogen's carbohydrate signature. So if we're talking about our cells, we mentioned this a little bit with our red blood cells, but all cells have different things on their surface of their membrane. So when our phagocytes recognize some of these things on the surface of a microbe's plasma membrane, it lets them know that it's foreign. So one downside is some of our microorganisms actually have a capsule surrounding them, so like this, and that can lead to our phagocytes not recognizing their carbohydrate signature because it's hidden beneath that capsule. So then our immune system does something else. It does something called opsonization. And it's pretty much where we take antibodies or other proteins that we call opsonins, and we coat the capsule of the pathogen so that we can use it to go in and bind to and basically phagocytize the organism still. So the second step, after we adhere, the phagocyte's able to adhere to the carbohydrate signature, or we undergo opsonization so we can adhere to one of those antibodies or opsonins. The next goal is to basically bring in the microbe into a vesicle. The vesicle that we bring the microbe into is what we call a phagosome. It's really great that we're able to bring in the microbe in this phagosome inside of the cell, but the phagosome doesn't contain anything to actually kill the microbe. So we have to fuse the phagosome with an organelle that does contain things that can break it down, and that organelle is our lysosome. So once we fuse our phagosome with our lysosome, we form what's called a phagolysosome. And then we can undergo and use those enzymes that are present in the lysosome to help acidify and digest whatever's inside that we brought in. Some of the things that we digest or bring in the phagosome are not digestible, so we consider those indigestible materials. We don't want to keep them in the cell, so we will actually exocytose those from the phagocytes. So again, here's a good little illustration. We can see the phagocyte adheres to the pathogen using some sort of receptor. So here's a little receptor that's going to cause it to bind to the microbe here, represented as the little green oval. We start to pretty much engulf or invaginate the plasma membrane of the phagocyte until we pinch it off and form a vesicle. The vesicle that's containing our microbe is what we call the phagosome. But again, there's nothing in the phagosome that can actually help break down the microbe that we brought in. So we have to fuse it with an organelle that contains the enzymes that are able to digest it, and that's the lysosome. Once we fuse the phagosome to the lysosome, we form the phagolysosome. And now we're capable of going in and degrading down whatever we brought into the cell. Some of the stuff we're not able to break down, it's indigestible, so we're actually going to refuse our vesicle that we made with the plasma membrane and exocytose any of that residual material. So phagocytosis is helpful for a lot of different microbes, but in some cases, pathogens that are brought in can't be killed by the lysosomal enzymes because the lysosomal enzymes basically turn the inside of the phagolysosome into a very acidic environment. Some bacteria and microbes are able to thrive in this acidic environment. So instead, we have to have our helper T cells help trigger the macrophage to produce what we call a respiratory burst, which is going to help kill pathogens that are resistant to the lysosomal enzymes. The respiratory burst includes things like releasing cell-killing free radicals, producing oxidizing agents such as hydrogen peroxide, and increasing the pH and osmolarity of the phagolysosome. So again, like tuberculosis, it thrives in a very acidic environment, but it's hindered in a basic environment. So if we increase the pH, we're making it more basic, and that's going to help kill the tuberculosis, or at least help prevent it from replicating very quickly because it's not in its ideal environment. And then, of course, we also have some defensins that are produced and released, especially if we're talking about neutrophils. And this was our antimicrobial protein that's capable of pretty much putting holes into a bacterial cell's plasma membranes, effectively lysing it. So those were our phagocytes, and our phagocytes were our monocytes and our neutrophils. Now we can move on to another type of cell, which is our natural killer cells. So these are non-phagocytic cells, but they're very large granular lymphocytes. Lymphocytes of the adaptive immune system are specific. These are not specific. And they basically act like the police of our blood and lymph. So they're constantly circulating around, looking for any intruders. They're capable of killing cancer and virus-infected cells before our adaptive immune system is activated. Because remember, our adaptive immune system is really slow and it takes a while for us to build up those defenses. So what do natural killer cells kill? They basically are going to attack cells that lack what we call self-cell surface receptors. So all of our cells in our body have a specific receptor on their plasma membrane to let them know that that's our own cell, that it's a good cell, don't touch it, it's fine. Any cell that doesn't have this self antigen on its receptor to let it know that it belongs to us is going to cause the natural killer cell to go in and attack that cell. So if it doesn't have the self receptor, then it's going to be attacked by a natural killer cell. And it kills by apoptosis, which is just programmed cell death. It's the same method as cytotoxic T cells, which we'll see later. But they're not really picky or specific, and the natural killer cells really just reflects the non-specificity of the cell itself. They're also capable of secreting really potent chemicals that are going to help enhance that inflammatory response. And moving on to inflammation. So inflammation is going to occur anytime we have any damage to our body tissues. It's a non-specific response, and it can include like trauma, intense heat, chemicals, or any type of infection. I will say that a lot of people think inflammation is a bad thing, but inflammation has several beneficial effects. So for example, if you have inflammation in a certain area, inflammation is going to help prevent the spread of whatever damaging agent is in that tissue to other tissues. So it's able to kind of help wall off that infection. It also is going to help dispose of cellular debris and pathogens, and inflammation is going to alert our adaptive immune system, and it's going to help set the stage for repair. So how do you know you have inflammation present? Well, there's 4 cardinal signs, and the 4 cardinal signs include redness, heat, swelling, and pain. And sometimes we consider impaired function to be a 5th cardinal sign. So for example, if you have an inflamed joint, say in your knee, that may hinder you from being able to use that joint, that's going to help force it to rest, which is going to help it heal. Okay, so stages of inflammation. When we're talking about the stages of inflammation, the first stage is really going to be where we have a flood of inflammatory chemicals being released into our extracellular fluid. And these chemicals are released by injured or stressed tissue cells. So for example, our mast cells are going to start to release histamine, which is a key component of the inflammatory response. We're also going to have vasodilation, and increased vascular permeability, as well as phagocyte mobilization. So let's talk a little bit more about the inflammatory chemical release. So chemicals can be released by those into the extracellular fluid by those injured tissues or immune cells. Like I mentioned previously, histamine being released from mast cells is a huge and crucial key inflammatory chemical. Other than histamine, we have some other things like kinins, prostaglandins, and cytokines. I don't know if you've ever heard of a cytokine storm, but all of those can help also initiate inflammation. And then if we're talking about a pathogen being the result of the inflammation, we have complement, which we'll talk about a little bit later. So all of these chemicals, including histamine, cause vasodilation of those local arterioles. If we have vasodilation and we're dilating those blood vessels, it's going to stretch the blood vessel walls, which means that we're going to be making those capillaries leaky. Making the capillaries leaky because we have those stretched walls is a good thing. And we can also attract phagocytes to that area. So here's a table of some of the inflammatory chemicals. I really just want you to know what the inflammatory chemicals are and what they do collectively as a whole. You don't need to understand the specific source of each one of these or necessarily the specific physiological effect unless we talk about it in more detail. So the second step of inflammation is we release all of those chemicals, and now we have vasodilation and increased vascular permeability. And we have that again because of vasodilation. We've stretched the cell walls, we've stretched our blood vessel walls, and we've increased the permeability because we've made them a little leaky. So vasodilation is going to cause a local hyperemia, which is going to increase the blood flow. And that's going to lead to redness and heat. And if you remember, redness and heat are two of our cardinal signs. The increased capillary permeability also causes exudate, which is basically fluid containing the clotting factors and antibodies is going to be leaked into the tissue spaces. As a result, we get local swelling because of that fluid leak. Swelling is also referred to as edema. And this swelling is going to help push on nerve endings, which is why we have pain associated with inflammation. So pain can also be the result from the release of toxins and bacteria, toxins from bacteria, or release prostaglandins and kinins. If you've ever taken aspirin actually inhibits prostaglandin release, which is going to help prevent this sense of pain. Some of the benefits of swelling or edema is that the surge of the fluid into the tissue is going to help sweep that foreign material into our lymphatic vessels. And we want it to be swept there so it can be processed by our lymph nodes. It's also going to help deliver clotting factors because, again, we have some sort of tissue damage and blood vessel damage, as well as complement to that area. And the clotting factors, like we talked about in chapter 17, they're going to form that fibrin mesh that's going to act as a scaffold for repair. And the mesh is really important that we created because it's also going to help contribute to isolating the injury so that we can't have movement of those microbes to other tissues. And the last thing is really phagocyte mobilization. So remember our phagocytic cells, we have neutrophils and macrophages. The first ones on the scene are actually going to be our neutrophils, and then are going to come our macrophiles. And there's pretty much 4 steps in which our phagocytes are mobilized to help infiltrate the whatever site of damage we have. So the first step is going to be what we call leukocytosis, and that's going to be the release of the neutrophils from the bone. So we're going to increase the number of leukocytes that we have anywhere from 4 to 5-fold of what we would if we didn't have any sort of tissue damage at all. How do we initiate leukocytosis? Well, those injured cells are going to release what we call leukocytosis-inducing factors. And in response to those factors, our neutrophils are going to start entering our blood from our red bone marrow within just a couple of hours. And the number of neutrophils, like I said, is going to increase 4 to 5-fold. And we're going to see an increase in white blood cells in general as well. The second step is going to be margination. So margination is where we have endothelial cells of our capillaries in the inflamed area are going to project what we call CAMs. And CAMs are just cellular adhesion molecules. These CAMs are really important because once they start projecting these CAMs, they're going to basically help stop neutrophils that are flowing by in our blood and cause them to kind of slow and roll along and cling to the blood vessel. And once we have that, neutrophils can also sprout CAMs on their surface once they're activated. The third step is diapedesis. So we have more chemical signaling basically prompts those neutrophils to help flatten and squeeze through the endothelial cells of the capillary walls. So the process of them leaving the bloodstream and entering into the tissues is called diapedesis. And the fourth step is chemotaxis. I mentioned this to you before in chapter 17, but chemotaxis is basically the method in which cells sense a concentration gradient of chemicals to help know where they're supposed to move to. So those inflammatory chemicals that are being released act as kind of like a signal for them to know where to go. So those neutrophils and other white blood cells will basically move to a higher concentration gradient of those signals, of those chemicals. So here's a good picture or an overview illustration where we have leukocytosis, or the production of 4 to 5-fold more neutrophils and white blood cells entering into our bloodstream from the bone marrow. Then we have marginization, where these cells are going to start sprouting what we call CAMs, or cellular adhesion molecules, that cause the neutrophils to kind of slow down a little bit and kind of stick, so that they're signaled that this is the site where they need to leave. Then we have diapedesis, where our neutrophils are actually going to change their shape so they can squeeze through the endothelial cell wall, so that they can leave our bloodstream and enter to the site of the tissue. Once they're in the tissues, they have no idea where to go, so they actually have to use what we call chemotaxis. So chemotaxis is where they're following a high concentration of chemicals that basically signals, hey, this is where the damage is, come here. So pretty much the goal of inflammation as a whole is to clear that injured area of the pathogens, clear it of dead tissue cells and other debris, so that we can set the stage for the tissue to come in and be repaired. Here's a good overview of kind of what happens. I recommend you kind of follow through this flow chart, starting with tissue injury. You can also see where we have the 4 cardinal signs of inflammation down here, and everything that kind of happens. I love these flow charts, so if you see them, I would definitely recommend going through them and kind of following along. Okay, something else that's involved in our second line of defense of our innate immune system is antimicrobial proteins. So these are going to help enhance the innate defense by, 1, attacking microorganisms directly, or by hindering their ability to reproduce. 2 really important antimicrobial proteins are what we call interferons, and these are proteins that are going to be released by virus-infected cells, or complement proteins, which is really just a group of blood-borne proteins. When they're activated, they lyse microbes via opsonization. So let's start with interferons. The interferons are a family of what we call immune modulating proteins. And what happens is that when we have cells that are infected with viruses, they're going to secrete something called IFNs, these interferons. And the interferons are going to warn the healthy neighboring signals that, hey, there's a virus around. How do they do this? They can enter into the neighboring cells. They stimulate the production of proteins that are actually going to block the viral reproduction, and they help degrade viral RNA. So we have 3 different types. IFN type A and B work this way, and we'll talk work this way, and we have interferon alpha and beta that also help activate natural killer cells. Our other type of interferon that's really important is interferon gamma, and it's actually secreted by lymphocytes, and it has widespread immune mobilizing effects because it's able to activate macrophages. So when we're talking about interferons, they can activate natural killer cells if they are alpha or beta interferons, and macrophages if they're gamma interferons, so they can also help directly fight cancer. We've used artificially made interferons to help treat disorders such as hepatitis C, genital warts, and multiple sclerosis. So here's a good picture to kind of explain what's going on. Here's a cell right here. This one right here. This is infected via virus. So here's our virus. Virus is going to come in, bind to a receptor on the surface of the cell, inject its nucleic acid. This nucleic acid is going to cause the cell that it infected to turn on its interferon genes. When it turns on its interferon genes, they're going to go through transcription and translation to produce the mRNA and eventually to produce the interferon molecules. So the cell that's infected will release what we call these IFNs or interferons. They're going to go and bind to neighboring cells. As you can see here, the interferon bound the neighboring cell to kind of warn them that there's a virus around. So how do they warn them? Once we have the interferon binding to the receptor, it's going to stimulate the healthy cell to turn on its antiviral protein genes. So we produce the antiviral mRNA and produce the proteins that can help go in and block viral reproduction. So the cell that was infected with the virus to begin with, we can't do anything for that, but we can help preserve and maintain the health of all the neighboring cells by using this interferon. The second thing is the complement system. And the complement system, like I said, is a group of blood proteins that are constantly circulating in the blood, but they're circulating in what we call an inactive form. And it's going to include proteins that are just numbered, C1 through C9, as well as factors like B, D, P, and some other regulatory proteins. Complement is pretty much going to provide a major mechanism for destroying foreign substances in the body. Once we activate complement, it's going to help release inflammatory chemicals that will help amplify all of the inflammatory process. And once we have that activated complement, it's also going to help lyse and kill bacteria and some other cell types that we have. It's non-specific defense system because we're still in our innate defense system, but it helps complement or enhance the effectiveness of both our innate and our adaptive immune system. So we have 3 different pathways of our complement. One is called the classical pathway, and the classical pathway is going to use antibodies. So antibodies are going to bind to the invading organism and then bind to a complement component, activating them. Once we have this double binding, it's called complement fixation. The second pathway is called the lectin pathway, and it's going to use lectins, not antibodies, to help produce the innate system, or produced by the innate system to help recognize those foreign invaders. The third pathway is alternative pathway. So the complement cascade is activated spontaneously, doesn't require binding of anything specific when certain complement factors bind directly to a foreign invader. So when we're talking about activation of these 3 different pathways, I thought I had a slide in here, I might later. I really just want you to understand all of these pathways, classical, lectin, and alternative, they all converge at the same endpoint. But what activates each one of the pathways? So classical, again, is going to be activated by antibodies. Lectin pathway is going to be activated by lectins. Alternative pathways activated spontaneously. That's kind of what I want you to get from the 3 pathways. And again, all 3 activation pathways are going to converge to something specific, which we call C3. So each pathway involves activation of proteins in an orderly sequence. Each step is going to catalyze the next, kind of like in a cascade fashion. And each pathway is going to converge. It doesn't matter, again, which one of the 3 pathways we take on complement protein C3. Once we activate C3, it's going to cause it to cleave into C3A and C3B. So we split C3. Once we split C3, it's going to enhance inflammation, promote phagocytosis, and cause cell lysis. So cell lysis begins when C3b binds to a target cell. When C3 binds to a target cell, it's going to cause this tunnel or pore kind of to be inserted into the plasma membrane of that cell. And that pore or that tunnel that's inserted is what we call the membrane attack complex or the MAC complex. So this is that hole I was telling you about before, being able to go in and pierce holes into a bacterial cell membrane. That hole that it pierces is actually this MAC complex. And because you put a hole or a pore into the plasma membrane of a bacterial cell, it's going to cause an influx of water, and ultimately it's going to cause lysis of the microbe. So that was one of the the components of the C3 that we split. That was C3b. C3b also causes opsonization. So opsonization, if you remember, we talked about it earlier. Basically, C3b can coat a microbe, and it acts like a handle for our phagocytes, so our neutrophils and macrophages, to help grab onto it, to help undergo phagocytosis in the event that we have a capsule around the bacterial membrane. So C3a is a little bit different. C3a and other complement protein cleavage products help amplify the immune response and inflammation. So C3a is going to go on to stimulate mast cells and basal cells to release histamine, and this is going to cause neutrophils and other inflammatory cells to be attracted to the site. So here's a really good picture. We have our 3 specific pathways. Classical, again, activated by antibodies. Lectin pathways activated by lectins. Alternative pathway is spontaneously. Doesn't matter which pathway we take, they all converge at C3, and they're going to cause C3 lysis into C3a and C3b. C3b can go on to cause opsonization. That's one of the things it can do. Again, so C3b is going to actually go and bind to the surface of pathogens and enhance phagocytosis. Or C3b can go on, option 2, and complex with other complement proteins, which you do not need to know the specific names of, to form that MAC complex or that pore or hole that's going to go into a bacterial cell membrane to cause its lysis. The other component of the C3 split, which is C3a, is a little bit different. C3a is going to go in to help enhance inflammation by doing things like stimulating histamine release, attracting phagocytes by chemotaxis, and increasing blood vessel permeability. Okay, antimicrobial proteins. We're still here. Let's talk a little bit about fever. So if we're talking about fever in response to some sort of invading microorganism, leukocytes in our macrophages that are exposed to some sort of microbe are going to secrete what we call pyrogens. And pyrogens are going to act on our body's thermostat, which is located in our hypothalamus, to help raise our body temperature. There are some benefits of fever because it's going to cause the liver and the spleen to help sequester iron and zinc. If our liver and spleen sequester iron and zinc, that's going to help inhibit bacterial growth because a lot of bacteria need both iron and zinc in order for it to replicate. Fever can also help increase the metabolic rate of tissue cells, which is going to help speed up the repair process if we have any damage.